Good afternoon, everyone. Welcome to the annual Wiley Prize in Biomedical Sciences Award and Lecture Program, which is kindly hosted here at Rockefeller University as part of the Friday afternoon lecture series. This year's prize will be the 14th award by our distinguished jury, Günter Blobel, Kai Salicati, Wayne Henderson of Columbia University, Robert Horvitz of MIT, Titia DeLong, who I think you all know, and Joan Stites of Yale University. This group worked as a team to make the selection of this year's award recipients from nominations submitted by leading research scientists. John Wiley & Sons continues to enjoy a productive and successful partnership with the scientific community for the advancement of research for over 208 years. The Wiley Prize in Biomedical Sciences was conceived as a way to recognize and to give back to that community. We chose to focus on biomedicine because of the extraordinary progress and change that continues to take place. Our goal is to recognize specific contributions or a series of contributions that demonstrates the nominees' significant leadership in the development of research concepts or their clinical applications. Particular emphasis is placed on research that champions novel approaches and challenges accepted thinking in biomedical sciences. This year's prize is being awarded to, This year's prize for the Wiley Prize in Biomedical Science is being awarded because of their advancement in DNA damage response. Having discovered that all cells can detect damage in their genetic material and respond in a way that ensures their survival. The winners are Dr. Evelyn Witkin, Professor Emerita of the Buckman Institute of Microbiology at Rutgers University, and Dr. Stephen Elledge, Professor of Genetics at Harvard University and Brigham and Women's Hospital, Boston. So I'd like to invite Evelyn and Stephen and Titia DeLong to come up and to accept the award, and then Titia will make the introductions. Thank you.
So it's now my great honor to introduce Evelyn Witkin and tell you something a little bit about her work. Evelyn did her first experiments in 1943, a time when classical genetics had matured, but it was still thought at that time that the genetic information was carried by the protein components of chromosomes rather than the DNA. This is pre-Avery. As a Columbia graduate student, she went to Cold Spring Harbor to try to induce mutations in E. coli, which at that time was a brand new established genetic system, following in the footsteps of plants, Drosophila. Evelyn's first seminal observation was that when she hit her strain of E. coli with UV light, the cells formed long filaments rather than doing their usual septation and cell division. In other words, in 1943, Witkin had discovered the E. coli equivalent of a DNA-damaged cell cycle checkpoint. She isolated the first checkpoint mutants before the structure of DNA was worked out. For the next 10 years at Cold Spring Harbor, first as a postdoc, and then as a staff scientist, you can see here with our very own late Norton Zindler, she studied the effects of UV on E. coli and isolated mutants. And many others did the same thing. This led to a bewildering diversity of UV-induced phenomena, including nucleotide excision repair, which had a different name at that time, changes in DNA replication, DNA mutagenesis, activation of dormant phage lambda, the Weigel effect, which I won't explain, and a delay in cell division. These outcomes of UV treatment were so dissimilar that they spawned separated subfields. And the idea that these were all related phenomena did not occur to anybody except for Evelyn Witkin, who had found in 1967, working then at Downstate, that phage induction and cell division arrest at the same UV response curve and timing, and importantly, were both required protein synthesis. This led her to suspect that these two phenomena might be regulated by a single pathway. Then in 1974, then at Rutgers, she looked into SOS replication. This was a term coined by Miroslav Radman, and showed that this error-prone DNA replication pathway also required protein synthesis for its induction by UV, again pointing to the possibility of a single regulatory pathway. And once Witkin realized what was going on, she organized a meeting in Gainesville, Florida in 1975, inviting 35 participants from different fields, the different subfields, to discuss the emerging evidence for a coordinated, inducible response to DNA damage and the central role of REC-A and LEX-A in this process. Then after this meeting, she published a truly remarkable paper, which I again read this morning on the plane back from London, in which she brings together data from close to 250 publications. It's a very diverse set of data, and proposes one coherent model for how the E. coli DNA damage response could work. That is the model that we now know as the SOS response. A pioneer woman in an almost entirely male field in those days, Evelyn Witkin is one of the most brilliant biological scientists of the 20th century, and I think we are all extremely fortunate that she was willing to not only come here to receive the Wiley Prize, but also to give a talk using a tool, PowerPoint, that's somewhat new to her. Evelyn, thank you for coming. Switch on your microphone.
Okay. Is it switched on?
Yes, it is on.
Okay. Thank you. If anybody had told me a year ago that I would be here today about to give a talk on E. coli's SOS response, I would have recommended a very good psychiatrist.
You don't want to run a computer.
Oh. I could have said no thanks, but on the other hand, the Wiley Foundation made me an offer I couldn't refuse. So here I am, and I hope that you can get in the mood for some ancient history, because that's the only kind I can talk to you about. And I'm going to try, as best I remember it, to tell you about how the SOS response in E. coli was discovered and how its scope and regulation were understood. I'm not going to show you very many slides, and the ones that I show will not be replete with all kinds of elegant molecular mechanisms, which I'm sure you are used to these days. But in the period when I was doing most of this work, in the 50s, 60s, and 70s, the approaches that we had to understanding ultraviolet mutagenesis were definitely not very molecular. We were interested in what happens in that first cell division after exposure to the radiation. And there was no way to get at the actual molecular events. What we had to do was try to use our ingenuity to infer the biochemistry from manipulations of the conditions in which we induce the mutations and especially by the use of mutants that disturbed the process. And we had to try to infer the biochemistry from that kind of data. I first encountered ultraviolet light in Cold Spring Harbor in 1943 when I was a Columbia University graduate student. I was there at Cold Spring Harbor for the summer to learn how to handle bacteria because with the approval of my advisor at Columbia, Theodosius Dobzhansky, I had decided I wanted to do my Ph.D. research with E. coli instead of with Drosophila as I had originally planned. And the new field of bacterial genetics was just taking off at Cold Spring Harbor, and I wanted to be a part of it. On my first day at Cold Spring Harbor, I reported to the director of the laboratories, Dr. Miroslav Radman, and he handed me a culture of E. coli and pointed me to an ultraviolet lamp and said, "Go, induce mutations." And on my very first experiment, I discovered a radiation-resistant mutant of E. coli, which then became the subject of my PhD research. What I found was that there was nothing particularly remarkable about the resistant mutant. What was remarkable was the sensitive parent strain. This was a wild-type strain. It happened to be the one that was used at Cold Spring Harbor, the B strain of E. coli. And it was extremely sensitive to ultraviolet light. And I found that even a very low dose of UV would cause an irreversible arrest of cell division. And these cells kept growing in long filaments. They looked under the microscope like a bowl of spaghetti after a while. And after about 3 hours, most of the cells would die. Now that, in retrospect, some years later, I realized that that was an extreme example of a delay in cell division that occurs in most bacteria and most organisms, actually, in response to DNA damage. And usually the delay is limited, and usually the cell division begins once the DNA repair is finished. But in this B strain, the arrest of cell division went on, and it was lethal. Actually, it seems that this was an extreme case of what is a much more limited cell division delay, which is part of what we now call checkpoint. The reason I'm mentioning this filamentous growth is not so much because it led us to think about the SOS response. It didn't at that point, certainly. But in a number of years later, I realized that the delay in cell division was a result of the action of an SOS gene. And it seemed to me a little bit spooky that my first beginner's luck mutation landed in an SOS gene that I would only appreciate 30 years later. And that made me think of a quotation from T.S. Eliot, which seems very apt in that situation. He wrote in 4 quartets, "The end of all exploring is to arrive where you started and know the place for the first time." It felt very much like that. Well, the first real step towards the SOS response was Leboeuf's finding in 1950 that ultraviolet light induced prophage in a lysogenic strain of B. megatherium. And soon after that, it was extended to other bacteria and other bacteriophages, and including a lambda phage in the K12 strain of E. coli. And that was a hint that ultraviolet light could turn some genes into action. The next step, I think, was the Wiley effect. Wiley, a few years, it was probably 1953, somewhere around there. Wiley found that if he irradiated a lambda bacteriophage and then infected E. coli, the irradiated phage, the fate of it, whether the amount of survival and the frequency of induced mutations in the phage, depended on whether the host cell, the bacterial cell, was also exposed to ultraviolet light. If the irradiated phage infected a non-irradiated host, the survival was very low and there were almost no induced phage mutations. But if both the host and the virus were irradiated, then the phage survival was very high and the survivors were loaded with induced phage mutations. That effect has had many names on and off over the years. I'm going to call it Foque mutagenesis here. And that was certainly a step on the way to the SOS response. It played an important part, actually, a few years later in defining the response. The next step along the way, I think, was 1967. I published 2 papers in that year that I think, in each in a different way, foreshadowed the SOS effect. The first paper was one in which I had noticed extremely provocative similarities between prophage induction and filamentous growth. And the similarities were very striking. Just I'll mention a few of them. They were both mass effects that affected every cell in the population. They both required the REC-A gene in order to be produced. Anyway, the parallels were very striking. They seemed to be co-regulated. And I made the suggestion in that paper that a bacterial gene that encoded a cell division inhibitor might be regulated in the same way as the prophage when the prophage is induced, and that there might be other bacterial genes that had repressors similar to lambda repressor. I should have mentioned that Luria in that year had just shown that when lambda prophage is induced, the lambda repressor is destroyed. So I thought that the bacterial gene encoding a cell division inhibitor might have a repressor similar enough to lambda repressor that it would respond to the same signal, and that other bacterial genes might also. Now, the next day after that paper came out in PNAS, I had a call from Francois Jacob, who asked if he could come and visit me in my lab. He did come, and the first thing he did was laughingly accuse me of being a witch. He then told me that a paper from his laboratory by him and a couple of his co-authors was about to be published in the very next issue of PNAS, which would strongly confirm the co-regulation of lambda prophage and filamentous growth. They were describing a mutant of E. coli called TIF, T-I-F, which without any DNA damage, it took no exposure to ultraviolet light or any other damaging agent, simply raising the temperature to 42 degrees, this strain would show both prophage induction, if it was a lysogenic strain, filamentous growth. They were both responding to the increased temperature in this mutant. So that was nice. Then my second paper in 1967 was on something else, but also foreshadows an aspect of the SOS response. And this paper was a kind of culmination of the 20 years or so that I had devoted to studying the mechanism of UV mutagenesis in E. coli. And I had earlier concluded that what causes an induced mutation is an unrepaired UV photoproduct, most likely a pyrimidine dimer, remaining in the DNA during the first replication after the irradiation. And what I proposed in this paper, first of all, I found in this paper that the mutation called LEX-A mutants, did not produce any UV-induced mutations. The strain was somewhat UV sensitive, and there were no induced mutations among the survivors. And I suggested in this paper that the LEX gene might control production of an error-prone DNA polymerase that was capable of replicating right past a non-coding lesion like a pyrimidine dimer. I suggested that it might be a new polymerase. At that time, we only knew of one DNA polymerase in 1967. We only knew the polymerase we now call Pol1. And of course, when we found there was a mutant that lacked that polymerase, and the cell made its DNA perfectly well, we realized there must be others. But at any rate, in this paper, I suggested an error-prone DNA polymerase being responsible for UV mutagenesis. Well, the next step was a very important one. 1971, a paper appeared by Defa, Foque, and Radman. It's the first time that Miroslav Radman enters the picture. And this paper suggested that, well, first of all, the thing that they did, the experiments they did, repeated and reinterpreted the Wiley effect. And they proposed that the LEX-A and REC-A genes control the appearance of an error-prone replication system that can replicate past and unrepaired lesion in the DNA. And immediately after that, a group in Paris, led by Jaqueline George, published a paper in which they showed that the TIF mutant that Jacob's group had originated earlier now also showed the Wiley effect at high temperature. This strain, without any UV exposure, without any DNA damage, expressed lambda prophage induction, filamentous growth, and the Wiley effect. Of course, it couldn't do prophage induction and filamentous growth in the same test tube, but if it was not a lysogen, it would do the filamentous growth. And it also allowed the irradiated phage to produce a lot of mutations without any DNA damage. Well, right after that, Miroslav Radman, who was then a postdoctoral fellow at Harvard, circulated a memorandum that he had written, a multi-page, very thoughtful piece, where he elaborated on this Defa paper. And this is the first time he coined the SOS part of the story, because he suggested that REC-A and LEX-A regulate the production of an error-prone replication system, which he called SOS replication. And it was, I won't go into the details of what that memorandum described, but it was a very thoughtful and very interesting piece. I read it with great interest because it seemed to me very likely that the SOS replication that he was proposing was very much the same as the error-prone DNA polymerase that I had proposed a bit earlier. At any rate, I wasn't very, the other point that Radman made was that he suggested that the same activity that's responsible for the Wiley mutagenesis in phage is also responsible for bacterial mutagenesis. Now, I was very interested, of course, in that because it seemed to me very possible, very likely, that the bacterial mutagenesis and the Wiley mutagenesis were carried out by the same kind of error-prone activity. First of all, viruses use the enzymes in the host, and why would there be two different error-prone polymerases, one for phage and one for bacteria? So it just seemed very likely it was one activity. However, there was no evidence in all the work I had done on UV mutagenesis over the years. I had not seen any evidence that that activity in bacterial mutagenesis was inducible. And I was anxious I couldn't see any immediate way of putting that to a test, but I thought that was important in order to be sure that there was just the one activity. Well, nevertheless, Radman and I were very excited that now we really each held a piece of a puzzle, and we could put them together and see where they led us. Because I had linked prophage induction and filamentous growth as co-regulated, and Radman and his group had linked Wiley mutagenesis and prophage induction as co-regulated. And so now we had three activities that seemed to be Rec-A and Lex-A dependent and consequences of DNA damage. Now that led us to begin to think about a broader kind of system, possibly. I had been working at that time on the properties of a mutant that lacked the DNA polymerase that we thought at that time was the only one E. coli had, now known as Pol1. This strain, I had been looking at UV mutagenesis in this strain, and it had a very peculiar quality. The strain was only slightly UV sensitive. It made the frequency of induced mutations was the same per survivor in the Pol-A mutant and in the Pol plus parent. So there was nothing remarkable there. But at very low doses, sublethal doses, the Pol-A mutant made 50 times more mutations than the Pol plus parent. And that was strange. And as the dose went up a little bit, the two curves came together more, and at a slightly higher dose, when the lethal effects begin to show, there was no difference between them. But I wondered why the burst of mutations at this very low dose in the Pol-A mutant. And I thought perhaps if the activity that's making the mutations is inducible, maybe the Pol-A mutant induces these SOS functions at a lower dose than the wild-type strain. And there was one way to look at that. Because I found in the literature that somebody had actually looked at the dose response of prophage induction and Wiley mutagenesis in the Pol-A mutant compared to the wild-type. And as you can see, this is a composite curve for both lambda induction and Wiley mutagenesis because they were almost indistinguishable. So this is just showing that curve. I have an arrow somewhere. Yes, here it is. It should be an arrow. Yeah, the top one. It's not. Okay, it's not working. Okay.
Push this button.
Push. This one, okay. I don't know, I couldn't get that one to work.
It's just very dim.
Okay. Yeah, this is the curve for the Pol-A mutant, lambda induction and Wiley mutagenesis combined. And this is the wild type combined curve for the two functions. And you see at the very low doses where the burst of mutations occurred in the Pol-A mutant, there's much more induction of lambda and Wiley mutagenesis than there is in the wild type. There's very little here. And if the activity for bacterial mutagenesis is inducible as an SOS function, and if this represents the dose response in the Pol-A mutant of SOS functions, then that could explain why there's such a burst of mutations at this very low dose and why it disappears as the two curves come together. I'll just leave this for the moment. I wanted to find a more direct and convincing way of showing that this bacterial mutagenesis is inducible. And I wondered if we could do that by using the TIF mutant, which, by the way, had since been mapped in the Rec-A gene and was now called Rec-A 441. Now, this mutant, Rec-A 441, the TIF mutant, could not be tested for the induction of UV mutagenesis, bacterial UV mutagenesis, in the original strain, which was a K12 strain. Because at the 42 degrees, where the SOS response is expressed constitutively, filamentous growth is lethal. So you would not be able to test for UV mutagenesis. So what I did was transfer the RECA441 gene into my strains, which were derived from my original radiation-resistant mutant, which I had called B-bar-R. And B-bar-R is incapable of delaying cell division. The mutation knocks out the inhibitor of cell division. And so I could transfer the RECA441 into my strain and then examine the way it handled UV mutagenesis at high temperature and see if that worked out. Okay. Here we see the very low doses. These are extremely low doses. Why am I not having luck with this arrow? This is the other one. Arrows don't seem to like me.
You got it. Did you have to?
Oh, yeah.
Make sure you point the right way.
Okay.
20 minutes.
Oh, okay. Oh, let's see. Okay, well, I have to do this quickly. I'm getting to the end of my time. All right, now, these are just the two temperatures. This is 30 degrees. These are very low doses of UV, almost no killing. These are sublethal doses. The temperatures didn't make any difference to the survival curves. But here you see, at 30 degrees, the TIF mutant and the REC plus, there's no difference at 30 degrees. The REC plus, given a heat treatment of 40 minutes after the UV, also shows no difference. But the REC441, given a 45-minute heat treatment after the ultraviolet exposure, shows this big jump at the lowest dose, about 50 times as many mutations. And gradually, the difference between the two curves diminishes, and when you get to this point in the survival, there's no difference, and for the rest of the dose curve, there's no difference. So this seemed to be better evidence, more convincing evidence, that the activity responsible for UV mutagenesis in the bacteria is part of the SOS response and is inducible. And it could be demonstrated in this material. Well, I'll have to go quickly through a few other things. Well, at this point, Radman and I were pretty energized the way things were shaping up. And we decided to examine the literature and look for potential SOS functions by looking for UV effects that had been described by others, which either required REC-A or LEX-A or both, and which required a period of protein synthesis following the irradiation. Well, we found quite a few of those, and I'll just show you quickly. This was just a quick look through the literature. I don't want to go through them all. I don't have time for that. But you can see we found quite a few potential SOS functions. I'll mention a couple of the ones that were most important. A regulator of exonuclease 5. Exonuclease 5 is responsible for DNA degradation at single-strand ends. And after UV irradiation, unless that exonuclease is controlled, that degradation is lethal. So that was an important one that had been shown to require REC-A and LEX-A. And the other one that I want to mention here, let's see, there was another one that I thought was important. Well, of course, here's our inhibitor of septum formation, which leads to the filamentous growth. Well, there was another one that I wanted to call attention to, but I'd better not. Anyway, as time went on, we developed more and more possible functions and documented them with evidence that they were, in fact, members of the SOS cluster of functions. And Radman wrote a very comprehensive paper about the SOS response in 1974, and he was able to list more of these. And then I wrote a review in 1976 where the list got even longer. And here I'll show you the way the list looked in about the early 80s when we had already identified a bunch of genes that could be linked to the functions. And these were all induced, SOS-induced genes, required REC-A and LEX-A and protein synthesis. And the list is getting longer. I would just like you to notice that a number of these functions, collectively, of course, they all promote survival and reproduction in some way or another. But many of them have an effect on increasing genetic variability. For instance, competence for transformation is increased. I don't know if this list shows it, but there was also movement of transposons is increased. And aside from the mutation rate going up, by the way, I didn't mention that when we found that the TIF mutant turned on UV mutagenesis at high temperature, our controls showed that at 42 degrees if you just incubate the strain at 42 degrees with no UV, it's a mutator. So the mutation rate, the error proneness, carries over to the replication of undamaged DNA. So there are a lot of things that, and there also are things on this list that show that ordinary repair, like excision repair, there it is. The genes that affect excision repair, although they're present at baseline levels that are that can do a lot of repair, all of them are greatly increased after damage. And so REC-A is hugely increased, and REC-A is involved in many kinds of important repair. So anyway, that was, we're on the way to understanding a little more about this system. And I should also point out the UMU genes. UMU, C and D are this is a little operon that is important, very important for UV mutagenesis, because it turns out it encodes DNA polymerase 5, which is the error-prone DNA polymerase that makes UV-induced mutations. And I must say that in Graham Walker's laboratory, Stephen Elledge was a graduate student at that time, and he had a lot to do with working out how these genes operate, and we owe a lot to that. I think that's all I have time for. I'll just show you my last slide. Oh, no, I wanted to show you two more, but very quickly. This is the way we understood the regulation of the system by the late 70s, and it's basically the way we still understand it today, with some, here we have, and there are about 43 genes that we know now that are part of the SOS system. And there are just a few of them here. What happens is, after UV, the arrest of replication that is caused by pyrimidine dimers or other lesions releases single-stranded DNA, and then with ATP and REC-A, you get a modification of REC-A, which is now called REC-A star. And what we know now about REC-A star is that a large number of REC-A molecules are cooperatively bound to a single strand of DNA, and in that condition, they have a coprotease activity, which results in the self-cleavage of LEX-A and also of UMUD, which doesn't show here. And basically, that's the regulation. These things are, and they're not all simultaneously induced, not all to the same extent. We know a lot more about the subtleties now, but that's basically the regulation as we now understand it. And I think this ought to be, oh, well, this is the, I'm sorry, I'll go back to this one quickly. This slide was made for me by Myron Goodman, who is the person who has illuminated the actual molecular steps involved in the error-prone replication at a pyrimidine dimer. And what they have shown, and he made this slide just for me to give at this talk, so I feel very flattered. Some of the information in this is quite new. He shows that Pol5, which consists of 2 units of the cleaved UMUD, so it's UMUD prime, which has been cleaved in cooperation with Rec star, combined with 1 UMUC, that's Pol5. If it combines with Rec A and ATP, I think you can see them in this, Pol5 mute is the actual error-prone complex that can do translesion replication opposite a pyrimidine dimer and either put in a mutation or not, depending on the details. But that's the actual error-prone DNA polymerase in action. And Myron has just showed that the enzyme has an intrinsic ATPase activity that ATP hydrolysis causes the Pol mute to dissociate from the DNA, and that prevents the mutagenic continuation of this error-prone enzyme, which is error-prone even without damage, to get off the DNA in a hurry. Okay, now my very last slide. This is Miroslav Radman and me in Paris about 2 years ago, having almost as much fun as we had chasing down the details of the SOS response. That's it. The screen? Can you get that off the screen? Mm-hmm. Okay, thanks.
I need to go back to my chair.
Thank you.
Thank you, Evelyn. So I will now introduce Steve Elledge. Sorry, I have to forward the slide. That's not Steve. I tried to make some timing link between Evelyn's days and Steve's days. And actually, he's from Paris. Where is it? Illinois. He's not born in La Paris. Okay, so like Evelyn Witkin, Steve Elledge contributed to our understanding of the DNA damage response not only by providing key data, but importantly by first intuiting and then proving how this pathway works, in his case, in yeast and mammalian cells. As Evelyn pointed out, Steve is a graduate student at MIT working on the SOS response, specifically on translesion polymerases, when he became interested in understanding whether eukaryotes had a similar SOS-like system. And as a young HHMI investigator at Baylor, he set up screens for yeast DNA damage response genes and then figured out how these genes worked. Early on, Steve had the idea that the eukaryotic DNA damage response might function as a signal transduction pathway, using a logic similar to growth factor signaling, with protein kinases becoming activated by a ligand, in this case damaged DNA, and then activating downstream protein kinases that would orchestrate different aspects of the DNA damage response. So Steve's real breakthrough came when he demonstrated that one of the DNA damage response genes he had identified in yeast was indeed a protein kinase acting in a signal transduction pathway that enforces cell cycle rest and guides appropriate DNA repair. Importantly, he then moved from the yeast system into mammalian cells where he again identified critical DNA damage response genes, showed how they transduce the DNA damage signal, demonstrated how one of the two main sensors of DNA damage, the ATR kinase, is activated, and worked out many of the downstream targets of the signal transduction pathway. His remarkable oeuvre has led the field for more than 2 decades and really forms the basis of our current understanding of how the eukaryotic DNA damage response works and its role in cancer. Steve has received many forms of recognition, many awards, but this is the first time he is honored together with Evelyn Witkin. The Wiley jury felt it was appropriate to recognize these two giants of the field who are working decades apart, have given us the fundamental insight that all cells have evolved the ability to sense damage in their DNA and then take the appropriate actions to guarantee the cell's survival with an intact genome. Steve, tell us your story.
Thank you. Okay. Well, thanks for that wonderful introduction, and it's a real pleasure to be here, and I want to thank the Wiley Foundation, and I'm especially grateful to be here with Evelyn Witkin, who, as you saw, really set the wheels in motion in understanding how the DNA damage response works in bacteria in a time when there weren't many tools. So they had to really do a lot of hard thinking and clear thinking. And I hope you all appreciated that, because I find that to be very inspirational. So I'll take you through my journey, and ironically, it intersects in a funny way with what Evelyn talked about. I just want to take you through, this is a normal karyotype of a cell, a little appreciation of the DNA damage response. And in a tumor derived from a mutation in a DNA damage response gene, you see that there's a lot of differences there. In particular, if you blow up these chromosomes, these colors represent different chromosomes, you can see they've been sheared and stitched back together. So this is very clearly important for human health. So I just want to give you a little background on the types of mutations. There are lots of types of DNA damage here that occur, and I just want to point out that some of them are more serious than others, like double-strand breaks and cross-links in DNA. But whenever there are lots of sort of standard repair mechanisms that recognize these common base adducts occasionally, or dimers, and remove them. But the real problems happen when DNA replication encounters these. Then you have a real problem, and you need something special. Now, the sources of damage are shown here. We have lots of sources: UV light, just the metabolism in your cell, reactive oxygen species, chemicals, and replication errors. And there are a lot of these. And this just shows you that in each of your cells per day, there are about 20,000 depurination events, another 5,000 oxidation events, methylation events. Your genome's under constant assault. In fact, during this talk, each one of you is going to incur about 3 times 10 to the 16th events. And I know this must make you very uneasy to think about that. And so I actually tracked down all the sources of these. And so if you can just avoid these, you'll be just fine. So just to take you to the beginning of my story on how I got interested in this, in work that I was doing in yeast, I call it a tale of 4 amino acids. And I had done work in the SOS response, as Evelyn mentioned, as a graduate student, but I fully intended to never work on it again. And not that I didn't like it, but I thought I should do something else. And I decided I was going to work on plants. And I went to Stanford to work in Ron Davis's lab. But by the time I got there, I figured out that plants weren't quite as exciting as E. coli, and so I looked for something else. And so I decided that I was interested in inventing gene targeting. I thought that was a really important thing, and I wanted to do it in mammalian cells. There had been some in yeast around that time. So the first thing that I decided to do was I wanted to clone the recombinases from mammalian cells. And that would be the equivalent of what you just heard about, REC-A, the E. coli protein. This is a Western blot of that. And so my idea was to get the mammalian version of that. But I didn't think I could necessarily go from bacteria, so first I thought I would look in yeast. And when I did that, I used antibodies to the E. coli protein, and I found a yeast protein, very clean blot, one protein band, and about the right size. So I was pretty excited. I decided to clone this gene using lambda GT11 technology. I isolated the gene. I could show that this was it. It encoded that protein. And then I did one more experiment because I knew that an E. coli REC-A was inducible. I looked to see if this gene was inducible. And sure enough, if you treat with UV light, the transcript for this goes way up. And I said, great, I got it. And the idea then was to take the yeast gene and through homology and hybridization, identify the mammalian gene, find that, purify the proteins, coat DNA with it, put it into mammalian cells, and target genes, knock them out. That was the idea, and then I would just see where this would take me. So that was the idea. So I cloned the gene, and I sequenced it, because you had to sequence it by hand back then, and I gave the sequence to a friend of mine, Andy Buckman, who actually had access to what was now must be the world's smallest database of DNA sequences, and he was going to run it against that. And he called me up and goes, Steve, I've got some good news and some bad news. And I said, okay, well, tell me the good news first. And he said, we identified your gene. I said, great. What's the bad news? He says, it's not REC-A. So there was a little problem, and the problem was 4 amino acids. And it turned out that REC-A, and the only homology it had with this gene that I cloned, were the last 4 amino acids. And this is just a cross reaction. And I proved that by mutating this and chopping the end up, completely obliterated it. So what was this gene that I cloned? Quite by accident. Well, it turned out to be the small subunit of ribonucleotide reductase. And that catalyzes the reduction of this 2' hydroxyl group in ribonucleotides to deoxys. So it's important for DNA synthesis. But this was nucleotide metabolism. And I can tell you, I did not want to work on nucleotide metabolism. I was really depressed. I didn't know what to do. It could only have been worse if, like, I'd cloned histone or something, right? Sorry, Dave. I know that I'll get back to you. But that was in 1985, remember? We didn't know anything. So I was stuck. I didn't know what to do. I was going to throw this stuff away. But maybe there was something good in it. And I remembered back to the fact that I did that northern blot. And I realized that this thing is really massively inducible. And at that time, yeast really was sort of on its own. And they were looking for some damage-inducible genes. They'd find things that were like 2-fold. But this is 20-fold. This is a huge effect. And so I realized that this must mean, based on knowledge of Evelyn's work, that there must be some SOS-like thing going on. And this is DNA replication. So I figured that there must be some way to sense DNA damage and transduce that signal to this promoter. So now I had a tool, this promoter, and I decided to try to work out the pathway of how this is regulated. So I took the RNR promoter and hooked it up not to LAC-Z, which is what most people were doing at the time, but to a gene called URA3. And the reason I chose URA3 is because you could both select for it and against it. So you could select for constitutive mutants because normally this is off in the absence of damage or replication stress. And you could look for constitutive mutants that were URA+, or if you tickled the cells with a little bit of stress, such as hydroxyurea, and turned this on, they became sensitive to a drug called 5-FOA, which is a toxin that gets metabolized by the URA3 gene. So you could select for uninducibles. And I'll show you the mutants that we got in our constitutive screen first. These are called CRT mutants. Three of them formed a transcriptional repressor complex that sits on the promoters of these genes. We later showed that in response to DNA damage, it comes off the DNA and turns on genes. Sounds like LEX-A. But most importantly, we found DNA polymerase mutants and also mutants in genes involved in nucleotide metabolism. And this really said that there must be something at the polymerase that's being sensed at the problematic replication forks. So we then set out to get the uninducible mutants. And I told you we tickled the cells with hydroxyurea. So I'll show you the plates here. So if you look at the top half, this is just yeast cells plated on media that lacks uracil. And you can see that these don't grow. Okay, this is wild type in this segment, and these are the damaged uninducible mutants that we selected, the done mutants. They don't grow. If you add hydroxyurea, wild type grows, but these can't activate URA3, so they don't grow. Now we do a similar experiment, now in the presence of uracil and 5-FOA. You can see in the absence of hydroxyurea, they all grow. But if you add a little bit of hydroxyurea, just a smidgen, the wild type cells are dead because they've induced URA3, but the mutants aren't. And this was the genetic selection. So what came out of this? Well, the first two genes were really quite informative. One was a protein kinase. I'll come back to that in a moment. And the other was another DNA polymerase, epsilon. And we initially thought this might be the sensor for damage. We now know that it actually houses associated proteins that are important for this response. But the protein kinase was critical because we hypothesized that it was a signal transduction pathway, but we got a lot of flack for that because at that time people thought signal transduction came from the outside of the cell to the inside of the cell, and what are you talking about? The key here is that a protein kinase could be required for this process, but maybe it's incidental. Maybe it controls the phosphorylation of something else that indirectly is involved, or maybe it responds to DNA damage. So we took advantage of the idea or the knowledge that a lot of protein kinases phosphorylate themselves when they're activated. And so we made antibodies to this protein. We could see that if you immunoprecipitate this protein from cells treated without or with a DNA damaging agent, methylmethane sulfonate, you can see that the protein incorporates p32. This is now yeast cells fed p32, so the place was hot as hell at the time. You can't see much of the protein here, but we overproduced it, and you can see here when it's overproduced, it's a much more striking effect. And importantly, two different mutants that affect the kinase mechanism block this. So we now had something to work with. We had a signal transduction pathway, and it led to the idea that there are signals which are distortions in the DNA or interference with replication forks, and there are proteins that are scouring your genome looking for these alterations. And when they find them, they set up sort of a solid-state signaling apparatus that activates signal transduction molecules that then contact effectors and carry out the physiological effects of the damage response in eukaryotes, including DNA repair, transcription, in mammalian cells, senescence and apoptosis, cell cycle control, as showed by Weinert and Hartwell, immune surveillance, and even tanning. That's right, when you go to the beach. So we worked out this pathway in yeast. These are the genes in yellow that came out from my lab, but many people contributed to this. And we found that there was like approximately four layers of proteins. There are these protein kinases that are PI3 kinase-like kinases, but they actually are phosphorylating proteins, MEK1 and TEL1, and then two intermediate adapter-like proteins we called mediators, and then a layer of other protein kinases called checkpoint kinases. Done ones down here. We found a master regulator of that, RAD53, and a variety of effectors. We went on to mammalian cells. We worked out a very similar pathway. And in response to double-strand breaks, the ATM, which is the TEL1, contacts mediators and checkpoint kinases to control these proteins down here. Replication blocks are controlled by the other half of the pathway, the ATR-ATRP pathway. And I'll tell you more about how this works in just a moment because we understand mechanistically how it gets activated and what it's sensing. I just want to point out that these genes are really important for controlling genomic stability and preventing tumorigenesis, and so all the ones in red have been linked to tumorigenesis in mammals, and in one way or another. So you can also probably see well-known tumor suppressors like the BRCA1 gene here. And this is, I drew this as more of a linear pathway, but believe me, it's a really a highly interconnected network. You can't really show it properly in a diagram. So how is this working? How is it sensing replication blocks? Well, if you have a lesion here that blocks replication, you have a replication fork, it'll stall there. You generate, the helicase continues to unwind, you generate single-stranded DNA, and that's covered by a single-stranded DNA binding protein called RPA, and you can see the similarities to what goes on in bacteria. And RPA now serves as a scaffold to start recruiting proteins. The first protein it recruits is ATR and this subunit ATRP that we cloned a few years ago. We found that ATRP actually recognizes RPA on single-stranded DNA and it localizes the ATR kinase there. A second sensor, the Rad17 complex, is recruited by a double-strand, single-strand junction here, coated with RPA, so that induces this protein to localize here. This is a clamp loader. It loads a pCNA-like clamp, shown here, called the 911 complex. So that loads on. It brings with it two other proteins, Rhino and TOP-BP1. And Bill Dunphy showed that TOP-BP1 could activate ATR. It turns out that ATR phosphorylates it, and then TOP-BP1 activates it. Then ATR then begins to phosphorylate a lot of substrates and sets in motion the signal transduction pathway. So this is really solid state. It's happening on the DNA. And it's a fail-safe mechanism because you have to have two independent sensory events to occur simultaneously in the same space. And this is to keep this process off unless you really have a problem in a replication fork. Because if you spuriously turn this on, you can induce all kinds of deleterious effects like the activation of the nucleases and things that helicases that you want to keep off until you need them at the right place at the right time because they're dangerous. And also this can induce apoptosis. Now, we found this protein kinase cascade, and we thought, well, you know, what are the substrates? We were interested in finding those. They should be critical. And we knew a few substrates, and we were able to use that knowledge in a sort of mass spec way to identify more substrates. And I won't go through the mechanism of how we did that, the methods, but we expected to find about 15 proteins whose phosphorylation is regulated in response to DNA damage, but instead we found thousands. And greater than 1,000 proteins are directly ATM or ATR substrates. And if you add in all the other kinases, it may go up to as many as 2,000. And these affect all kinds of different processes in cells. Other stress response pathways, chromatin, ubiquitin ligases. A huge number of them are involved in controlling splicing and metabolism, carbon metabolism. It rewires how the cell works. Transcription, a number of different processes. So this is really quite surprising to us, and what this says is that the DNA damage response is really having a profound alteration of the cellular physiology. And it's critically important that cells get this right. If there's really threatening damage, you have to solve that problem or you have to get out of town. So a variety of, if you just look at the types of proteins that are phosphorylated, you can see they're involved in many different pathways, proliferation, protein trafficking, signal transduction, various developmental processes, and a large group of them are involved in DNA replication. So in the last few minutes here, I thought I would take you through something new that we're doing that has to do with another aspect of the DNA damage response. And that's shown on this slide. So the DNA damage response, as I said, controls a number of pathways that are relevant to tumor suppression. They promote proper replication and some DNA repair, certain types of repair, not all of them certainly. The sensory mechanism can activate the right ones at the right time. It promotes genomic, or inhibits genomic instability by controlling cell cycle transitions coupled with proper replication. It can activate immune surveillance or apoptosis or something called cellular senescence. And cellular senescence is a major tumor suppressor pathway. It basically induces the differentiation of a cell. It causes the cell to permanently leave the cell cycle, and a number of things happen to it. They flatten out, and they start secreting lots of cytokines to alert the immune system. And this turns out to be an important tumor suppressor mechanism. I'll tell you more about that in a moment. But first, I want to talk about another aspect of senescence, and this is the two-edged sword of the DNA damage response. So senescence is a tumor suppressor mechanism, but it's also been linked to aging. So a very interesting experiment was done in the Van Dersen lab a few years ago, published in Nature, where they took a rapidly aging mouse. This is a hypomorphic allele of the BUB-R1 kinase. It's a spindle checkpoint kinase, so it throws off lots of aneuploidy. And these mice have all kinds of rapid aging diseases. They age almost twice as fast as a normal mouse. And you can see this poor little creature is messed up. And when they look at its cells, they can detect which ones are senescent by a stain, and they accumulate a large number of senescent cells. Now, the clever experiment here is that Van Dersen's lab knew that there was one gene that gets induced in senescent cells. They set up a conditional lethal situation where they could kill the senescent cell. Okay, the cells accumulate, but they can snap their fingers and make them go away. And what you can see is that when you get rid of the senescent cells, now the mice don't age. And I just want to say that this was done in an aging model, but it's been repeated now in normal mice, and it does seem to be affecting aging and longevity. So that's a pretty important thing. So what's the pathway? What do we know about the senescence pathway and the DNA damage response? And this is some work that's from Tizia DeLong's lab here. We know that senescence signals, which are short telomeres or massive amounts of DNA damage or activated oncogenes, can induce this pathway. And we know a lot about the cell cycle arrest part because it's controlled by the two main tumor suppressor pathways in mammals, the P16 pathway, which inhibits RB, and the P53 pathway, which induces the CDK inhibitor to turn off CDK2 and CDK1. So these are both a DNA cycle-independent kinase inhibitors. But there's a third branch. And the third branch is important because when they talk about the aging model of these mice, they don't refer to it as a cell autonomous effect. They think there's a cell non-autonomous effect. And this branch of this pathway, which is completely independent of P16 and P53, activates this, what they call the senescence-associated secretory phenotype, or SASP. And it throws a lot of cytokines out of cells. Now, initially, it made sense because you might think if you have a damaged cell, you might want to alert the immune system to come. So these are pro-inflammatory cytokines. And it's also true, actually, that and these are all independent pathways, interestingly enough, but they all do very similar things. But if you're infected with a lot of viruses, actually activate the ATM and ATR pathway because when they're replicating, they look like damage. And this may affect their function as well. So the critical question here is how do you activate this branch? How does ATM and ATR turn this on through NF-kappa B? How does that work? And I won't go into the details, but we've recently discovered an important part of this switch, which is that a transcription factor called GATA4 gets stabilized. Now, GATA4 gets turned on about 4 or 5 days after senescence signals. About the same time P16 gets turned on. P53 is much earlier. So somehow a few events must happen before this switch is flipped. And we know in part how it's flipped because this isn't transcriptionally induced. It actually becomes stabilized, and normally it's destroyed by autophagy. And by this autophagy adapter P62, and somehow ATM and ATR shuts that off. This accumulates and activates a cytokine, IL-1A, that is in a feed-forward loop with NF-kappa B, so it flips this switch. Now, people have shown that if you have senescent fibroblasts and you mix them with tumor cells, they make those tumors grow a lot faster. So the idea here is that as you develop this inflammatory phenotype, it promotes aspects of aging and also cancer. So it's a two-edged sword here. So I just take you through the fact that this protein becomes stabilized and aged as we age. And this is a mouse. We've taken tissue, this is the mouse liver, and looked at GATA4 levels. And you can see in older mice, you get massive induction of GATA4 at the same time as you get P16 induction. So this is true of all the tissues that we've looked at. And we were also able to get some extracts from human brains, and we see a very similar effect. You can see that in young brains, GATA4 is very low, but in brains of older individuals, it actually goes very high, as does P16. And so it looks like brain cells may be undergoing a form of senescence. And it's not clear what that means for a brain cell. The significance of it with respect to brain function is not at all clear. But we do know that these these cells that are, this is now immunofluorescence of these cells of neural tissue in young and older brains. You can see that there is a serious number of these nuclei are turning on this pathway. Maybe it's and co-localizing. The same cells are, for the most part, but not exclusively, turning on both P16 and GATA4. And so I think one of the exciting things about this is that that now we have the ability to switch off the SAS part of this pathway. We have conditional mice now, and we can begin to ask questions about how important this branch of the senescence pathway is to human aging and also tumor suppression. So I just want to leave you with the idea that DNA damage is really information that the cell needs to know about. And once it knows about this, it makes a lot of decisions. Just like E. coli makes decisions, mammalian cells, in a much more complex way, that uses different pieces of the puzzle, but senses it in a very similar general mechanism. It takes this information and it makes decisions about a variety of different events. So these are all controlled by the DNA damage response, including meiosis, and even various aspects of development are critically regulated by the damage response. So I'll just stop here and thank you all for your attention. Thanks.
That was well planned. My prince.
It wasn't a glass slipper.
I think since it's 5 o'clock, I think we should allow time for some questions. And I would ask both Evelyn and Steve whether they're willing to take some questions.
Sure.
Yes? So, questions for the two Wiley speakers? Maybe I can start by asking both of them, maybe Steve first, why the mammalian DNA damage response is so different in its wiring, in the way it reacts to DNA damage than what you see in E. coli.
Why?
Why did we evolve?
Yeah, that's a good
new
question. Why?
system?
Would you allow me to speculate on that? Well, first of all, I would just say that we are not as evolved as bacteria. So I know this is hard to take to, but they're much more highly evolved than we are, and they can do things in the shortest ways possible and get it just right. So they have fewer proteins. They do what they need to do. We're slightly more complex organisms. We have lots of different decisions to make. And I think that because there's so many different proteins and events that occur that have to be coordinated, it had to become a more flexible, modular sort of system. And I guess that's the way I look at it.
Can I ask a follow-up to that question? So, are there homologs of more of the bacterial proteins in the mammalian cells, but they're doing something else? Or have they sort of vanished and been replaced?
That's a good question. So there are homologs of course, Rec A, even though I didn't find it. You know, I wanted to find it, I didn't find it. It does seem to do something different. It doesn't mean that it doesn't have a contribution, because we have some evidence, but it could be very indirect, that something involved in recombination may maintain a damage signal over a long period of time. So I guess the way I look at it is that the different events that occur, you have to know when you're finished. And there is a handoff between RPA and recombination. You might want to sense that. We don't have any evidence that Rec-A homologs, RAD51, are being sensed in that way, but there's some things floating around that might point in that direction. But the rest of the system is very different. But like I said, for Lex-A, which is a repressor, we have the CRT1 complex. It gets turned off. It gets turned off in a different way, but it comes off the DNA. So, I mean, the sort of general principles are kind of conserved, but all of the details are really different. And it must have evolved separately, I think.
Yes, yeah.
No. It was all inducible at the level of transcription, yeah. Although there is some stabilization of some of them, and there's actually also regulated nuclear localization in response to damage. It's really a complex, interwoven, highly regulated thing, because apparently nucleotide levels are really critical. If you have too much of them, you mutate spontaneously, and we could talk about why that is. But no, it didn't have that.
Cat4 seems like it's bad for aging. So if you look how Cat4 in mice, does it live longer?
Yeah, so the question was, GATA4 seems like a bad thing for aging. If you knock it out, do they live longer? The answer to that is definitely no. And they don't live longer because they need GATA4 to make a heart. And so they're heartless. So they die embryonic lethal. It's odd that you use the same thing for a different purpose, but as you know, transcription factors really depend on the cell type that they're expressed in.
Okay. Well, I'm going to end this by thanking both speakers for a spectacular afternoon, and by thanking all of you for coming, and thanking the Wiley Foundation for having and supporting this prize. Thank you very much, Debbie. And